Welcome back, everyone. So far in our videos on ANOVA, we've been talking about scenarios where we've only been testing the influence of one factor at a time. But one of the great things about ANOVA is its ability to test multiple factors at the same time. So in the ANOVAs we've been talking about so far, we've been considering the influence of only one factor on our response variable at a time. And this is what's referred to as a one-way ANOVA. So for example, the effects of 4 different diets on the weight gain of lambs is testing the effect of diet on lamb weight gain. So we've got one categorical predictor with 4 different levels predicting our response variable, which is a continuous measure of weight gain in lambs. So this is a one-way ANOVA because we're testing only one categorical predictor in this analysis. But ANOVA is extremely flexible and can allow us to test the effects of multiple predictors at the same time. So if, for example, we were interested in testing the effects of both diet and housing arrangement on the weight gain of lambs, then this would in fact be a two-way ANOVA because we're testing the influence of 2 different predictors, diet and housing arrangements, at the same time. So let's say we've got this example where we've got 3 different diets and 2 different housing arrangements, lambs raised either with siblings or without siblings, and we randomly assign lambs to one level of each of these 2 factors, then we would get a scenario that looks something like this. With 3 different diet treatments and 2 different housing treatments, we've actually got 6 different combinations of possible treatments. And this is what's referred to as a factorial design, where we're collecting observations in all possible combinations of our 3 diet levels and our 2 housing levels. In this example, let's say we assign 10 individuals to each of these 6 different treatments. So now we've got 6 different treatment combinations arranged in this factorial way, where we can test 2 different null hypotheses at the same time. The first null hypothesis is that all of the diet means are equal to one another. And the second null hypothesis is that the housing means are equal to one another. As an example of this two-way ANOVA, a PhD student of mine, Andrea Meals, tested the influence of fish pheromones and water temperature on the tail spine length of these invasive spiny water fleas. Now, other cladocerans like Daphnia had been shown to respond to fish cues or chemicals of fish in the water called pheromones. And in Lake Michigan, water temperature was associated with increased predation risk. And so Andrea hypothesized that both pheromones and water temperature might influence the length of this protective tail spine. So Andrea raised Bythotrephes in warm and cold water, either with or without fish cues in this factorial way, and then measured the length of the spines in their offspring. And this is what she found. Here you can see the length of spines in control Bythotrephes and Bythotrephes exposed to fish cues in either warmer water or colder water. And you can see that there's not really any difference between control and fish pheromone Bythotrephes, but there was a difference between the warmer water and the colder water treatments. So ANOVA is a very flexible technique that can allow us to test the effect of any number of factors or categorical predictor variables on some continuous response measure. So ANOVAs can be generalized then to look at more than one categorical variable at a time. As examples of two-way ANOVAs then, we can test how caffeine and pollinator species affect visitation rates to artificial flowers, or how pesticide exposure and predator cues affect the survival of tadpoles. So let's say we were interested in testing the influence of both pesticide exposure and predator exposure on the survival of tadpoles. And we did this in a factorial way where we had 2 predator treatments and 2 pesticide treatments, where we had individuals assigned to receive one of these 4 different combinations of predator and pesticide treatments in a factorial design. So here we've got 4 different combinations. So why wouldn't we simply analyze these 4 different groups in a one-way ANOVA with 4 different groups? Well, the reason we don't do this is because our null hypotheses are specific to particular comparisons. These differences among the 4 groups in a one-way ANOVA doesn't allow us to specifically test our biological hypotheses about the influences of predators and pesticides. Instead, we want to explicitly test the effects of predators and pesticide exposure through a two-way ANOVA, where we look at the effects of predators and pesticides separately. And this approach then acknowledges that there are consistencies among these treatments. These 2 treatments in the reddish color represent the absence of predators, while those in the greenish color represent the presence, and the bars on the left represent the water treatment, and the bars on the right represent the exposure to our pesticides. So the idea here in a 2-factor ANOVA is that these aren't just 4 arbitrary groups that we want to compare, but there are logical connections between these groups that allows us to test explicitly these 2 biological hypotheses at the same time. So once we acknowledge that these groups are connected in specific ways, we can draw some specific inferences from the patterns in the differences among groups. So here I've used what's called an interaction plot to depict both the effects of pesticide treatments corresponding to the 2 different columns of observations and predator exposure corresponding to the 2 different colors. And so what we can infer from this is that survival looks to be higher overall in the water treatment compared to the pesticide treatment. And the tadpoles in the absence of predators tend to have higher survival than those in the presence of predators. So connecting the group means in these logical ways allows us to reveal patterns that wouldn't be so obvious when we were just making comparisons across the means as though they were 4 independent groups. Now, these are just hypothetical data. When we actually look at the data from this experiment, they look something like this, which is really quite an interesting pattern. In the absence of predators, shown in the open circles here, there doesn't seem to be any effect of the pesticide treatment at all. The control tadpoles had the same survival as those experiencing the pesticide treatment. But it's only in the presence of predators that we start to see an influence of the pesticide treatment. So how can we then explain these patterns? Is there an effect of predators or not? Is there an effect of the pesticide treatment or not? Well, we can see from this plot that the effect of the predator treatment really depends on the level of the pesticide treatment that we're talking about. And the effect of the pesticides really depends on whether or not predators are present. And this is what's referred to as a statistical interaction. The effect of one variable depends on the level of the other variable. And one of the really powerful features of testing multiple hypotheses at the same time within a multiple factor ANOVA is our ability then to test the effects of each of those factors individually, while at the same time testing for their interactive effects. That is, asking the question, does the effect of pesticides depend on whether or not predators are present? And so an interaction effect then is an effect of one predictor variable that depends on the value or level of some other predictor variable. And testing multiple factors within a single ANOVA allows us to test for the presence of interactions that we can't test for if we were to have tested these effects in two separate one-way ANOVAs. Now we can perform multiple factor ANOVAs in R using this AOV command, where we predict survival by both pesticides and predator treatments. In addition to these additive effects of the influence of pesticides and predators individually, we can also test for their interactive effect by specifying the interaction level by a colon in between pesticides and predators. So this model here tests both the individual effects of pesticides and predators in a two-way ANOVA, as well as the statistical interaction between pesticides and predators by specifying the interaction term with a colon in between these two variable types. Now we can specify the exact same statistical model using a little bit of a shorthand in R with the asterisk symbol. When we specify two predictors connected by the asterisk symbol, what we're saying is that we want both the additive effects of each of these two things individually, as well as their interaction term. So both of these two models will perform the exact same statistical analysis, and the second notation is just a little bit of a shorthand of the first notation. So here are the results of an experiment then that was designed to test the effects of grazing by limpets by either excluding limpets from being able to forage in an area or allowing them to forage in an area using experimental enclosures, while at the same time testing the effect of where that grazing occurred, either at the low tide line or midway between the low and high tide lines. And in this case, the response variable was the biomass of marine algae on that particular surface area. So this is then a two-way ANOVA where we're also testing for the presence of an interaction between the effects of herbivores and the location of the herbivory. So here are the results of our statistical test, where we can test the influence of herbivores on its own, the effect of height or location on its own, as well as the statistical interaction between these two variables. And what we can see from this result is that there is an overall effect of herbivores on the biomass of marine algae, but more importantly, there's a strong interaction between the effects of herbivores and location. And what this tells us is that the effect of herbivores depends on the location that we're talking about. And we can depict this using our interaction plot, where we can use lines to connect the experimental means in our herbivore excluded treatments and our herbivore presence treatments at either low or mid tide lines. So you can see from these crossing lines then that the effects of herbivores depends on whether we're talking about the low tide location or the mid tide location. In the low tide location, there was a very strong effect of herbivores on the biomass of algae. When herbivores were excluded, there was a lot more algae than when herbivores were present. But in the mid-tide location, there was very little effect. If anything, there was a positive effect of the presence of herbivores on the biomass of algae. So this two-factor ANOVA then allowed us to test the influence of herbivores and location on the biomass of algae, but also to test the statistical interaction between these two factors that tells us that the effect of herbivores is not constant. It depends on which location we're talking about. Now, statistical interactions can get quite complicated. And so if you're ever in a pickle for explaining what a statistical interaction means, remember this basic description that when we have a statistical interaction, the effect of one factor is not constant. It depends on the level of the other factor. So in this plot below, we have a statistical interaction between water depth and whether or not eggs are exposed to UVB on their survival. And so if we're not sure how to explain this interaction, we can simply begin by saying the influence of one factor is not constant. It depends on the level of the other factor. So the effect of UVB on the survival of toad eggs is not constant. It depends on what water depth we're talking about. Now, in an interaction, we can also interpret the results in the completely complementary way. And that is the effect of water depth on toad survival is not constant. It depends on whether those toad eggs are exposed to UVB or not. So in particular, the toad eggs shielded from UVB exposure had consistently high survival, whereas the toad eggs exposed to UVB had high survival at deep water depths, but at shallow water depths, their survival was compromised. So when in doubt, in the presence of a statistical interaction, interpret the interaction as the effect of one factor is not constant. It depends on the level of the other factor. Now, statistical interactions can provide us with really powerful tests of biological hypotheses. If you remember that as a master's student, I was interested in the potential for dietary protein to constrain reproduction and growth of young mice. I'd made the observation that the growth rates of deer mice varied a lot from one year to the next. And I hypothesized that in some years, mice had access to lots of high protein food, which allowed them to grow quickly. But in other years, their growth was constrained by their availability of high protein food. And this allowed me to make the complex prediction that mice supplemented with food would grow faster because of this constraint on their ability to grow because of their lack of dietary protein, but that we would only see this effect of supplementation if the natural levels of food was low. In a year where high protein food was abundant, we wouldn't expect the supplement to have any sort of effect. And this is in fact what I found. I performed my dietary protein experiment in 2 years. In 1996, the growth of control mice and high protein supplemented mice were identical. But 1996 was a year of naturally high growth rates. In the next year, growth rates were much lower. But the provisioning of high protein food allowed the mice to continue to grow quickly, even though something in the natural environment prevented the mice without access to high protein food from growing quickly. This interactive effect suggests then that the availability of high protein food differed between 1996 and 1997 because the supplementation of high protein food enhanced the growth of mice in 1997, but didn't enhance the growth of mice that were already growing quickly, presumably because they already had access to lots of high protein food in 1996. So statistical interactions then allow us to perform more sophisticated tests of our biological hypotheses. So let's walk through an example of this. Let's say we had the hypothesis that Cisco eat more large zooplankton than small zooplankton because the larger zooplankton are easier to see. So we might perform an experiment then where we have tanks of zooplankton either with fish or without fish. And we might make the prediction that the average size of zooplankton at the end of 3 days will be smaller in the aquaria with fish than in the aquaria without fish, because the fish are eating the larger zooplankton. Now, this might seem like a reasonable prediction given our hypothesis, but remember the details of the hypothesis. What is the mechanism of this hypothesis? In our hypothesis, the mechanism for why we expect to see this pattern is because the larger zooplankton are easier to see. Now, if we satisfy this prediction and find that the fish have eaten all of the larger zooplankton, is this in fact because the larger zooplankton are easier to see, or is it because of something else? Maybe because of optimal foraging that means that it's more beneficial for fish to eat the larger zooplankton. If we want to test this specific mechanism of why Cisco eat larger zooplankton than smaller zooplankton, we need to get a little bit more sophisticated in our experimental design. So in addition to having tanks with and without fish, we could explicitly test this biological hypothesis by also having tanks where the fish weren't able to see, by making the tanks dark. So we can then test a more sophisticated prediction that the average size of zooplankton at the end of 3 days will be smaller in aquaria with fish than aquaria without fish when the fish can see. But when the fish can't see, we would not expect there to be any difference in the average size of zooplankton between tanks with and without fish. So now we've got a two-factor experimental design, tanks with and without fish, tanks in the presence of light allowing the fish to see, and tanks in the darkness where the fish cannot see. And our prediction then is fundamentally about an interaction. We expect the effect of fish on the size of zooplankton to not be constant. We expect this effect to depend on whether or not the fish are able to see. So in this case, we predict no effect of the presence of fish in darkness, but for the average size of zooplankton after 3 days to be smaller in the presence of fish when the fish are able to see. So in this case, we can use a factorial design and a statistical interaction to more directly test our hypothesized mechanism to get at our biological hypothesis more explicitly. So in a two-way ANOVA then, an interaction term tests whether the effect of factor A depends on the level of factor B. If there's a significant interaction, then the effect of factor A is not constant. It depends on the level of factor B. If the interaction term is not significant, then the effect of factor A does not depend on the level of factor B, and we can interpret then that the effect of factor A is constant. Finally, these interaction plots are extremely useful for visualizing the effects of interaction in a multiple factor ANOVA. So in a two-way ANOVA with two factors, A and B, there are two different ways in which we might find effects in the absence of interactions and ways in which we might find effects in the presence of interactions. So in this case, with our interaction plot, we can see that there's no difference between A1 and A2, but there is a difference between B1 and B2, where our B2 treatment has higher levels of our response variable than our B1 treatment. But this effect of B is constant. It does not depend on the level of A. So in this case, we would see a significant effect of B, no significant effect of A, and no significant interaction. Similarly, we might find that there is an effect of B. The blue dots are above the red dots. There is also an effect of A in this situation. The A1 dots are higher than the A2 dots, but the effect of A and the effect of B are constant. They don't depend on the level of the other factor. So in this interaction plot, we would have an effect of A, an effect of B, and no statistical interaction between A and B. In contrast, we might have a scenario that looks like this, where we have no overall effect of A. These dots aren't consistently higher than these dots. No consistent effect of B. The red dots are not consistently higher or lower than the blue dots. But we do have a statistical interaction where the effect of B depends on whether we're talking about A1 or A2. The blue dot is higher than the red dot in the presence of A1, but the blue dot is lower than the red dot when we have our factor level A2. We can also get statistical interactions that look like this, where the blue dots are higher than the red dots, and there's no difference between A1 and A2, but there is a statistical interaction. We don't have crossing lines here, but the effect size, the difference between the blue dot and the red dot, differs between our A1 treatment and our A2 treatment. So in this case, we would have no effect of the A factor, we would have a significant effect of the B factor, and we would have a significant interaction between A and B. In these interaction plots, we can interpret whether or not there's a significant interaction by whether or not the lines connecting our treatments are parallel or not parallel. In some cases, the presence of a statistical interaction will be indicated by these crossing interaction plot lines, but not always. In some cases, the lines can diverge, like the bottom plot here, which doesn't indicate crossing lines, but does indicate that the lines are not parallel to one another. So here we have different scenarios in the interaction plots that can indicate either the presence of a statistical interaction or the absence of a statistical interaction. So in summary then, a two-way ANOVA lets us test the effects of two factors at the same time. Randomly assigning replicates to treatments for each of these two factors in all possible combinations represents what's called a factorial statistical design. When we have a factorial design where all combinations between two factors are present in our statistical analysis, we can test not only the independent effects of the two factors, but also test for their statistical interactions. When we have a significant statistical interaction, this means then that the effect of one factor is not constant. It depends on the level of another factor.